Hi all. So today, these are going to be the introduction notes then for our start of our heredity genetics section in this unit. I wanted to start off first though with a few review points about meiosis. So ultimately, like you had in one of the questions from last class, was, what is the ultimate outcome of meiosis? Well, meiosis is absolutely necessary for sexual reproduction to occur in order to create those 4 genetically unique gamete cells with half of the normal amount of genetic information, so that when you have fertilization come together, you are going to have the appropriate number and consistent number of chromosomes in the next generation. A few vocabulary terms. First one, for homologous chromosomes, please make sure then that you know that this is going to be a pair of chromosomes. You inherited one from mom and one from dad. Also, I want you to know that if you see this X-shaped structure, this is actually a duplicated chromosome. And that each half of this then is going to be one of those sister chromatids. So together, they're going to make up each half of this duplicated chromosome. Now in this case here, crossing over has already happened, so these 2 chromatids are not anymore completely identical to one another, but they were at the start of meiosis 1. Last thing is that this right here, you can call this a chromatid, but you can also call it a chromosome. So they essentially are interchangeable and they mean the same thing. These X's are not quite a chromosome, they are actually a duplicated chromosome. So in this cell we would have 4 chromosomes, in this one we actually have 8 because both half of the X would count as a chromosome. So after the end of meiosis 1, we create cells then that have half of the number of chromosomes, because what we have done is we've separated those homologous chromosomes. So now these cells are haploid, and that between meiosis 1 and meiosis 2, the difference here is now we go from 4 chromosomes to 2 in this scenario, because now the sister chromatids separate from one another. So these cells are still haploid, and now these final gamete cells have half of the genetic information needed to start the next generation. So when we think about then meiosis and those genetically different gamete cells, some things to keep in mind as well as to how that genetic variation happened. It's due to the independent assortment. So when those homologous chromosomes separate from one another and rearrange themselves in meiosis 1, that can contribute to 8 million different possible combinations. And then think of fertilization, that only increases the amount of variation, because both mom and dad can create those 8 million possible combinations. So together it's like 70 trillion combinations of unique offspring. And then add to that, crossing over, really the amount of variation is exponential in terms of now we have those genetic recombinations of those chromosomes. So importance of meiosis as well, besides retaining chromosome number, is also that genetic variation. So for Mendelian genetics, we are going to be going over how you can identify the inheritance of genes through Mendel's laws, and we're going to be focusing on that today. But we'll also then make sure that we focus on DNA and RNA and how that relates to the inheritance of different traits, and that the genetic code is something that's shared between all living systems, and that we read our genetic information in the same way. So the behavior of chromosomes during the cell cycle allows for heritability. So this heritability then being able to create those genetically unique gamete cells and pass them on. So we have to start when we're talking about genetics with Gregor Mendel. Gregor Mendel is the father of modern day genetics, but he didn't go out to study the laws of inheritance. What he went out to study was he wanted to figure out how to make the best pea plant hybrid. So at this time, farmers were really into making hybrids, both plant variety as well as animal variety. And what he wanted to do was he wanted to try to figure out the best farming techniques in order to how to breed these hybrid plants. So if we think of a hybrid, you know, a hybrid's a combination of two different organisms. So if we think of, you know, cars, you can have hybrid cars, gas and electric. Or mythology, you can have a hybrid organism, half man, half horse. But in this case here, the pea plants that he worked with had different traits. And these pea plants, the different traits allowed them to be a really good model organism. But also, pea plants, they have a really quick generation time. It doesn't take a whole lot in terms of cost to be able to make lots of peas. And they tend to grow in vines, so they're not going to take up a lot of space either. So speaking of those traits, though, for these organisms, the other really nice thing about them as model organisms is that the traits were very objective. So the flowers were either purple or white, but not like a light purple. The plants were either tall or dwarf, but not a medium height. So he's also fortunate in that aspect to use peas as his model for what we now have as the laws of modern genetics. All right, so he started out his pea plants with pure parents. So for pure parents, it means that whenever they would self-fertilize, they would only and ever create that one variety. So if we think back to some vocabulary terms that you might be familiar with, it's like they would be the homozygous individuals. And when he had two different parents for the same trait that were both pure or homozygous, and he crossed them together, in the F1 generation, or that first generation or first file generation, he noticed something interesting. So in this case, he noticed that all the F1s were only purple. He didn't have any white. But when he took one of the F1s and he allowed that plant to self-pollinate, then he ended up with these ratios that were just about 3-to-1. So when all of his 7 different traits in the first generation, one of those 2 traits from the parents disappeared, and in the second generation, he ended up with this approximately 3-to-1 ratio. So a quick review of terms. Genotype, those are the genes or the alleles that the organism has. We represent those as letters. Phenotype is the trait that the organism shows, so we represent that as the kind of descriptive term, or what the phenotype, what the physical trait is. So there's homozygous dominant, where you only have dominant alleles, heterozygous, where you have a mix between the two, so this would be like hybrid, and then your homozygous recessive, only the recessive alleles. And then we have dominant and recessive traits in Mendelian genetics. And what it means that have dominant traits is that those individuals that have a dominant trait have at least one dominant allele. Dominant alleles are ones that can hide or mask a recessive allele. And that's really most of the time the case because they have some type of functional gene that's going to code for either some type of regulating factor for a protein that's made or the protein itself. So in this case, if you have brown eyes and you have the gene, the dominant gene, to code for melanin, which is going to produce that brown pigment. If you have blue eyes and you do not have a functional gene for melanin, the gene that you have inherited is not going to create that proper protein. So when we think about meiosis as well, because we are creating those haploid cells, what we know today is that those 2 homologous chromosomes separate. And so that in each one of our gamete cells, we are only putting one of the 2 alleles for each trait that we have. Gregor Mendel didn't know anything about chromosomes or DNA, but he still was able to logically reason that those plants had to have only one of these factors for each of their traits in their gamete cells. So for what we know then today, the gametes only are going to receive one allele for a given trait. And because of this, we call it the law of segregation. The parent that was hybrid When they make their gamete cells, they split those two alleles into different gamete cells. So a quick run through then of certain questions. You should be able to identify, what does true breeding mean? What is the genotype of a hybrid? What does the F1 refer to? What is the cross described here in the F1 hybrids if they were allowed to self-fertilize? E, when looking at the data presented, what ratio is it closest to? And then F, what is the dominant phenotype, short or tall, and how do you know? We'll go over these in class to start tomorrow. All right, another concept then that you should be familiar with is if we're thinking of dominant and recessive and that law of dominance, and that it is that one trait that can hide or mask the other. But if we think about the parents themselves, both the parent had a purple flower and the F1s also had a purple flower. So were those F1s, or if you were to see a flower, a pea plant in general in nature, would you know, is it homozygous dominant or heterozygous? Well, you could determine that if you complete something called a test cross. A test cross is where you're going to breed the dominant phenotype, which has an unknown genotype, it could be homozygous or a heterozygous, with a homozygous recessive individual. And that way you would be able to determine the genotype of that unknown dominant parent. So what you would do, again, notice in both scenarios they're crossed with a homozygous recessive, that if you end up with offspring that only resemble the purple parent, then chances are that the original parent was homozygous dominant. But you could know with certainty that if there is at least one of those offspring that are white, then that purple parent had to be heterozygous because they had to be able to give that recessive allele to their offspring. So again, we'll go over this question then tomorrow. Last concept then for today is this of independent assortment. So for independent assortment, remember that when those homologous chromosomes line up, before they are separated, they are going to arrange themselves in an order with each one of those, for us, 23 pairs of chromosomes arranging themselves in a different way. So maybe the first chromosome has mom on the right, dad on the left. Second chromosome has dad on the left, mom on the right in terms of the chromosomes that they gave you. And they can assort themselves in any of those independent fashions. So Mendel also reasoned that the inheritance of one of those factors over the other, they were independent of each other, that they didn't have influence on one another. So you could have a parent plant that was tall and purple flower, and then when they make their gamete cells, if they were heterozygous for both, one of the gamete cells could be tall and short, one of the gamete cells, purple and tall, one of them could be purple and short, one of them could be white and short, one of them could be white and tall. So you have these random combinations. So independent assortment, the segregation of unlinked alleles have no influence on one another. And so we'll go over how to calculate then the proportion of those numbers when you are looking at two traits together. So what I would like for you then to do for today is I want you to get started on the Mendelian genetics practice problems. I want you to do at least 1 through 10. If you want to keep going and you remember this, please go ahead. I'll be looking over your work then for today. But again, what I want you to do is 1 through 10. I will finally be in class tomorrow. So I can go over with you any of the questions you've had from the week. I have followed along, and it looks like you are doing well on your own and having a good understanding of the content. So again, for today, if you can just do the Mendelian practice problems 1 through 10, and I will see you tomorrow. Bye, all.